Welcome to this week's BioBite. This week we're going to talk about things that are neither plant nor animal. This includes many of our single-celled organisms along with protists and fungi and viruses. Our first stop will be viruses. Viruses are different from living cells. They are acellular. They're not actually cells. They have no metabolism and cannot grow. They depend on host cells to make copies of themselves. And in this, they are the ultimate parasite. They cannot replicate unless they're in a host cell using that cell's machinery. Their origin is uncertain. They seem to have collected pieces of nucleic acid from various sources, can infect nearly all life forms, and they are not only species specific, but tissue specific as far as what they will infect. Did viruses devolve from living cells? Did nucleic acid molecules escape from cells? Did they begin as self-replicating molecules? No one's sure. We don't know how viruses evolved. So there are many different types of viruses and many different kind of formulations, how they look, how they act, what they infect. Each virion has a nucleic acid, which might be a double-stranded DNA or a single-stranded RNA. They have a protein capsid, and some virions also have an envelope that is similar to a cell membrane. The next thing we're going to talk about is prokaryotes. We've talked about this a little bit before. Prokaryotes obviously are cells that do not have membrane-bound organelles. So they don't have the nucleus, they don't have the mitochondria, all of that. And prokaryotes are very important to us in microbiology. So first thing we're going to talk about is the history of microbiology. The size of prokaryotic cells led to their being undiscovered for most of human history. In 1546, Italian physician Girolamo Fracastoro suggested that disease was caused by unseen organisms. Prior to that, it could have been caused, their thoughts were that, a miasma in the air would cause the disease, any number of things, but they weren't thinking about unseen organisms causing it. So to study microbes, there are two technology strands that kind of allow us to do that. One is microscopy. If we didn't have microscopy, we couldn't see the microbes. We wouldn't be able to visualize them. And then, of course, infectious disease investigations. And there's a whole level of science and technology that goes into that. Antony von Leeuwenhoek was the first to observe and accurately describe microbial life. He is the first one to really have created a functional microscope and to see cells. And now we have modern electron microscopes that allow the study of cell substructure because we can see so much more with those electron microscopes than we can with the simple light ones. And then you have Louis Pasteur. We know him from pasteurization. He basically is the one that said if you raise it to a high enough temperature for long enough, then you kill enough of the germs that it's not going to be a problem. He refuted the idea of spontaneous generation, that the thought that living cells would just miraculously appear from non-living matter. And then you have Robert Koch, who studied anthrax. The causative agent is Bacillus anthracis. And he proposed 4 postulates to prove a causal relationship between a microorganism and an individual. Basically, how do we prove that this microorganism is causing a disease? So first, we say the microorganism must be present in every case of the disease and absent from healthy individuals. The putative causative agent must be isolated and grown in a pure culture. So we have to isolate that microbe and grow it in a pure culture. And the same disease must result when the cultured microorganism is used to infect a healthy host. So we culture that microbe, we infect a healthy host, and if it gives us that disease, then we know that is a disease-causing microorganism. Then we have to isolate that same microorganism from the diseased host. All of that tells us that that microorganism is disease-causing. Not all microorganisms are disease-causing. Some don't cause any diseases at all. They're all around us all the time. They're in our guts. Those microorganisms in our guts are what allow us to digest a lot of our food. But to see if one is disease causing, you have to go through these 4 postulates. And we still use a lot of these today. That's, the basis for a lot of our epidemiology. So talking about prokaryotic diversity, they are the oldest, the structurally simplest, and the most abundant forms of life. They've been around for billions and billions and billions of years. Over a billion years before eukaryotic life became a thing, the prokaryotes were there. 90 to 99% are unknown and undescribed. Less than 1% cause disease. They fall into two domains, the bacteria, also called the eubacteria, and the archaea, formerly called archaeobacteria. Many archaeans are extremophiles. They live in extreme environments. So prokaryotes differ from eukaryotes in that they are unicellular. Prokaryotes are single-celled. Only eukaryotic cells can be multicellular. Prokaryotes may stick together to form associations and biofilms, but they are still single-celled organisms. They're just colonies. The Cyanobacteria nostoc commune, it's unicellular, but it sticks together to form large clumps of this colonial mass. And you see it here in Oklahoma sometimes. I'll find a picture and put it in our class. But people eat it. It's kind of weird looking. But to think that that large clump of gelatinous goo is actually many single-celled organisms together is kind of interesting. So the cell size is another thing where they differ from eukaryotes. Size varies tremendously in prokaryotes, but they're generally much smaller than eukaryotes. They're usually less than 1 millimeter in diameter. They have a nucleoid region. The prokaryotic chromosome is a single circular double-stranded DNA found in the nucleoid region of the cell and often has plasmids. Cell division and genetic recombination. Most divide by binary fission. They don't go through the mitosis process that eukaryotes do, and they exchange genetic material extensively. So those are some of the ways they differ from eukaryotes. So getting in here, talking about genetic recombination. It occurs through horizontal gene transfer. No sexual reproduction happens. It's not a form of reproduction. Lack the internal compartmentalization that prokaryotes have, or that eukaryotes have. They have no membrane-bounded organelles, no internal compartments, and their ribosomes differ from the eukaryotic form. Their flagella are simple in structure and different from eukaryotic flagella. And their metabolic diversity, they have oxygenic and anoxygenic photosynthesis, and some are chemolithotrophic, meaning they can use the energy stored in the bonds of inorganic molecules. That's what we talk about with some of the extremophiles, where they are living in high sulfur environments, so they are using sulfur, some of those different things. And bacteria and archaea differ fundamentally. They differ in 4 key areas: their plasma membrane, their cell walls, their DNA replication, and their gene expression. Archaea have evolved to live in some extremely, harsh environments around, underwater volcanoes, things of that nature. So they have evolved a little bit differently than their bacterial cousins. Next, we're going to talk about protists. Protists is where we start kind of getting into eukaryotes, okay? The complexity and diversity of protists make them kind of difficult to classify. They can't be classified as plants, animals, or fungi. They could be split into as many as a dozen different kingdoms. Most of them are unicellular, but not all. Many of them have an amazingly high level of structural and functional complexity. Amoeboids and ciliates possess unique organelles, such as contractile vacuoles. So they have some really unique and unusual structures. And general biology of the protists, most protists are free-living. Some are parasitic. Asexual reproduction is common. Sexual reproduction may occur when conditions deteriorate. And some life cycles are very simple. Some are extremely complex. They vary in size from microscopic algae and protozoans to kelp more than 200 meters in length. So there's a huge variety of how large they can be and what shapes they can take. Ecology of the protists. Protists are of enormous ecological importance. Their photoautotrophic forms produce oxygen and function as producers in both freshwater and saltwater ecosystems. They're a major component of plankton, which are the organisms that are suspended in the water and serve as food for krill and any filter feeding animals and heterotrophic protists. Many protists are symbionts. They range from strict parasitism to mutualism. Coral reefs are greatly aided by symbiotic photoautotrophic protists in the tissues of the corals. So, how did we get from prokaryotes to eukaryotes? Well, one of the things that we think might have happened is that an ancestral prokaryote had undergone a membrane proliferation and then compartmentalization of cellular function into a nucleus, lysosomes, and an endoplasmic reticulum, and the establishment of endosymbiotic relationships with an aerobic prokaryote, and in some cases, a photosynthetic prokaryote to form the mitochondria and chloroplast. So this picture shows the hypothesized process of endosymbiotic events leading to the evolution of chloroarachniophytes. The resulting organelle is a plastid in modern chloroarachniophytes. The resulting organelle became a plastid in the modern version of this cell. So phagocytosis. This is basically how the little single-celled organisms eat. They don't have a way, they don't have a stomach, anything like that. They engulf a food particle, digest it using enzymes within a lysosome, and then send the undigested portions out of the cell through an exocytic vesicle. Transportation. Protists use various methods for transportation. You have paramecium that wave little hair-like appendages. You have amoeba using lobe-like pseudopods to anchor itself to a solid surface and pull itself forward. And then you have those flagellates that have a flagellum that they use a whip-like tail to propel themselves. So this diagram just shows a proposed classification of the domain Euchara. Currently, we're divided into 6 supergroups. Within each supergroup are multiple kingdoms, and dotted lines indicate suggested evolutionary relationships that still remain under debate. We're still figuring all of that out. We are technically eukaryotes, so are land plants. They kind of think that we all go back to a common prokaryotic ancestor. So next, we're going to talk about fungi. There are a ton of fungi out there. The kingdom contains about 80,000 species. They're mostly multicellular eukaryotes that share a common mode of nutrition. They're heterotrophic. The cells release digestive enzymes and then absorb resultant nutrient molecules. Most are saprophobes. That means they grow on dead things. Some are parasitic. Some exist in mutualistic relationships with plants or other organisms. Lichen. Lichen is a good example of a mutualistic relationship with a fungus and a photosynthetic bacteria. So the biggest organism on Earth is a fungus. They evolved from protists, kind of like we did. Molecular data tells that animals and fungi shared a common ancestor after plants evolved. So animals and fungi are more closely related to each other than to plants. Flagellated unicellular protist was likely our most common ancestor. We're still, puzzling all of that out. So a description of fungal structure best applies to the zygospore fungi, sac fungi, and the club fungi. That's, saying that we have fungal structure that we can look at and describe. The AM fungi, or arbuscular mycorrhiza, exist only as mycorrhiza in association with plant roots. So it can't really describe the fungal structure there. Fungal anatomy doesn't lead itself to becoming fossilized. They're squishy. and soft, and they tend to rot away entirely. They probably evolved a lot earlier than the earliest known fungal fossil, um, because mycorrhiza are evident in plant fossils long before that. So some points to remember from this one: viruses, what are the characteristics? What are the differences with living cells? The history of microbiology, Leeuwenhoek and his contributions. Prokaryotes, characteristics of bacteria, archaea, then protists, looking at the characteristics. What is endosymbiosis? Modes of transportation, phagocytosis, and then, of course, characteristics of fungi and how do they reproduce. And that's our BioBite for the week. Thanks. See you next week.